Alrighty, we're going to go hopefully pretty quickly through cells in this particular video. So first thing I want you to know is, generally speaking, in high school, when they talk about two types of cells, they mention plant and animal cells. What's important is that we're going to back up and talk about the two main types of cells that are bigger picture than that, which are prokaryotic cells and eukaryotic cells. Prokaryotic cells are things like bacteria and archaea, and that's this little guy off over here. Those cells are small. They don't have a nucleus. They don't have any of the things that you guys tend to remember from high school, like the mitochondria is the powerhouse of the cell. They don't have mitochondria. In fact, they're about the same size as the mitochondria, which is something that I'll bring back up again a little bit later on. Eukaryotic cells, on the other hand, they are bigger. You are made out of eukaryotic cells. They do have the bits and pieces that you think of that you learned in high school. They will have things like the nucleus, the mitochondria, which incidentally, mitochondria is plural. Mitochondrion is singular. You can't see the mitochondria in this picture. They have stained it with a fluorescent stain, and they're using a fluorescent or a microscope to be able to see that stain. And so what you're seeing is the cytoskeleton in this picture. You've got actin filaments and you've got microtubules. You don't normally get to see the cytoskeleton, but in this picture you're getting to see that. The cytoskeleton is what helps to maintain the shape of the cell, the size of the cell. It helps move things around within the cell. So those are the main two types, prokaryote and eukaryote. Now, the cell types that they tell you about in high school are plant and animal cells, and both of those two types of cells are eukaryotic, because plant cells, like the ones in this picture, they have what are called membrane-bound organelles, like the nucleus. They also have mitochondria, just like your cells do. They have chloroplasts, which are all those little green things that you're seeing in this picture. All of those organelles have this membrane around them, and only eukaryotic cells have those. So plants and animals, eukaryotic. Fungi, like mushrooms, those are also eukaryotic. And then protists, whether you think of them as their own little world or you realize that taxonomically they are plants, animals, or fungi, they have eukaryotic cells as well. Bacteria and archaeans. Now, you've maybe never heard of archaeans before. Those are things that live in really extreme environments. Think hot springs as an example. What you're looking at right here, these are little clusters of spherical bacterial cells, which for some reason my little pointer doesn't want to draw on it where I can see it, but that's two cells that I have circled right there, and they're really, really tiny, and that's bacteria. That could be like, well, that could be like gonorrhea as an example. Okay. Even your cells are so small that you're going to usually need a microscope in order to be able to see them. Because cells don't have a color to them, you're going to need a stain to be able to see them. So your cells aren't normally blue, but in the lab we tend to add things like methylene blue or eosin or hematoxylin, and we add those stains so that you're able to see them. This is one of the typical microscopes that we use to be able to see them. You'll get more on microscopes if you end up taking the lab portion of this course in person, or a simulation if you are doing things online. Okay, what I want you guys to know about cells is cells are essentially little protein factories. Their job is to make proteins, fold them into correct shapes, and then use those proteins to do other things. The instructions for how to make proteins are found within the nucleus of the cell. That would be this whole entire purple thing right here. In the nucleus, there's a structure that is called the nucleolus. That's right there. The nucleolus makes ribosomes, which are these little red dots that you can find out here. I don't know why it's making such a big zigzaggy line, but whatever. The ribosomes are the factory that makes the proteins based on the instructions that the nucleus sends it. So the nucleus, when you think of it as the brains of the cell, it has all the instructions to make everything the cell needs to be able to make, and the ribosomes carry out those instructions so that it can make the proteins. Then we have two different kinds of what's called endoplasmic reticulum. We have rough endoplasmic reticulum, which looks kind of like sandpaper when you look at it under the microscope, and that's because of all the ribosomes on the surface. And then we have smooth endoplasmic reticulum, which is all kind of through here. It looks smooth because it doesn't have ribosomes on the surface. There's a lot of overlap between what the rough and the smooth endoplasmic reticulum do, but to separate them for you guys, what I'll say is, and what this picture says, is the rough ER folds the proteins to give them the correct shape they're supposed to have. If you remember from the chemistry chapter, proteins have to be folded in the exact correct shape or they are not going to work. And we even talked a little bit about denaturation in that chapter, which is a misfolded protein, essentially. So the rough ER helps to fold that protein and give it the right shape. The smooth ER, on the other hand, doesn't have the ribosomes, and so it can't make the proteins and then fold the proteins. Instead, it makes things like lipids. So some of your lipid-based hormones like estrogen and progesterone and testosterone, those are made here in the smooth ER. It makes phospholipids so it can make more membranes. That's what this is doing off over here. Now, back to proteins. We have folded some proteins, we have made them. After that, we are going to ship our proteins off to this pink, I don't know, usually books like to call them stacked pancakes. Personally, my pancakes don't look like that, but whatever it looks like to you, man. So this pink stuff is called the Golgi apparatus or the Golgi complex. I don't care which way you like to call that. The Golgi apparatus, as it's labeled in this picture, it likes to modify proteins. So sometimes proteins can have carbohydrates attached. That makes it a glycoprotein. Sometimes proteins can have lipids attached to them. Sometimes we want to break the fold and fold it a little bit differently. The Golgi apparatus can do that too. So when it says it modifies proteins, it's adding things or it's folding it a little bit differently. That's changing the function of the protein in some cases. Then it packs the proteins in these little bubbles that are called vesicles, and those bubbles are the shipping containers of the cell's world. So because the Golgi apparatus can modify proteins and then put them in these little shipping bubbles, it is sometimes called the post office of the cell because it is what packs them in the shipping bubbles and then ships them wherever it's supposed to go. Maybe the protein's supposed to go to the mitochondria, it'll ship it to the mitochondria. Maybe it's supposed to go out of the cell, it'll ship it out in a secretory vesicle, like in this case right here, and then spit it out from the cell. That is called exocytosis or secretion when that happens. Now, some of the bubbles get extra special proteins, and so the name changes based on what's in that bubble. If the protein that's in the bubble has a digestive function, the bubble is called a lysosome, and it is now essentially sort of like a stomach for the whole cell. It's going to help digest things inside that cell. So there's a special function that white blood cells can do that is called phagocytosis. If this were a white blood cell and it met some of that gonorrhea from earlier, this white blood cell could phagocytize or eat that gonorrhea, and then the lysosome would digest that gonorrhea. So the lysosomes help to eat things like bacteria. Or if a mitochondrion goes bad, which can happen, they expire, a lysosome will autophage or autophagocytose the mitochondrion and then eat it and recycle some of the components. And so lysosomes are helping to maintain homeostasis inside the cell by eating things that have gone bad or were already bad and got into the cell. We also have other special little bubbles that contain enzymes that can produce peroxides, like hydrogen peroxide. Those also play an immune function. Some bacteria have evolved a way to get out of lysosomes, and so in that case we'll just dump peroxide on them and try to kill them with peroxide. That's just another little tool that we have inside of our cells to try to destroy things. So that's the difference between a lysosome, which eats, and a peroxisome, which essentially tries to poison something that's inside of our cell. Okay, plasma membrane. All cells have a plasma membrane. This is not unique to animal cells. Plant cells, fungal cells, prokaryotes, eukaryotes, every single cell on the planet has a plasma membrane. Every single cell on the planet has cytoplasm. That is not unique to us. And then all cells are going to have DNA of some type. It's just that sometimes the DNA is inside a nucleus and sometimes it's not. So those three things that I've circled, all cells have those. Some cells will have additionally, I'm going to flip to this one, a cell wall that is outside of the cell membrane. So this thin green line inside is the plasma membrane. Outside of that, we also have a cell wall in plant cells. So you can see that labeled right here if it wants to play nice and not draw an extra line. So they have both, and they've labeled them right beside each other in this picture. I like when they label them side by side. But you come back here, no cell wall. That's one big difference between animal cells and plant cells. All the other things that are labeled up here on this picture, they help ship the proteins around within the cell or maintain the shape of the cell. So microfilaments, those are things like actin filaments, which were like, if I come back to this earlier picture that was a long time ago now, and I probably should have just not gone back to it. Those green lines, those were the microfilaments that it's mentioning here in this picture that now I'm going to come back to. The intermediate filaments you couldn't see in that picture. That's really when you think about the cytoskeleton being like your skeleton, that's the intermediate filaments. The microtubules up here, I want you to think of those like they are railroad tracks or a highway. They don't move, generally speaking, but proteins get shipped over those so that things can move around within the cell. And then the centrosome builds and maintains the cytoskeleton as a whole. When it's time for the cell to do the cell cycle, and do mitosis or meiosis, the centrosome can tear down the cytoskeleton and then build it back up so that we can make more cells. So that's like the briefest version of kind of what cells are. Two big versions, prokaryote, eukaryote. And then within the eukaryotic world, we've got plants which have a cell wall and chloroplasts, and animals which don't have a cell wall and don't have chloroplasts but do have lysosomes. Hopefully that helps you throughout this chapter.